Human life would be impossible without gene regulation. Liver cells, for example, must express genes which are not expressed in brain cells or white blood cells. The genes which are expressed during fetal development must be those different from those expressed as an adult. The human genome includes thousands of transcription factors, whose binding to DNA controls which genes are on and which are off. Most human transcription factor genes belong to only several gene families, such as the helix-turn helix, Hmg box, homeodomain, helix-loop helix, nuclear receptor, and zinc finger families. Each of these gene families may include hundreds of separate genes of the human genome and be divided into smaller groups. For example, helix-loop helix, or HLH proteins, include genes required for neural and muscle development in the embryo, and others which interact with harmful toxins or components of cigarette smoke. There are hundreds of zinc finger proteins in the human genome, including many of the proteins which regulate fetal and embryonic development. Nuclear receptors, a subfamily of zinc finger proteins, include receptors for estrogen, testosterone, stress hormones, the glucocorticoids, thyroid hormones, and vitamin D. The first transcriptional regulators discovered belong to the helix-turn helix group. In addition to gene family members such as forkhead, whose homologs are important in development, and BRCA2, whose mutations can cause breast cancer, there are two large subfamilies, the Hmg box and the homeodomain. The Hmg family includes the SRY gene, located on the Y chromosome, which begins the process of gender differentiation in male mammals. Homeodomain genes include many which are involved in the differentiation of tissues during development, and are often considered as developmental master switches, such as PAX genes essential for early eye development. One interesting group, the Hox cluster genes, provide a molecular positioning map along the length of the body and limbs to inform cells of their location, so that they can differentiate correctly.